Thesis committee 7 years ago. So Peter obtained his PhD from Harvard under David Mumford in 1993, and he has had positions in Yale, and most recently he's a full professor at Columbia University. And what I can say about Peter is that his papers have already become classics, right? In illumination, in face recognition, in physics-based vision, and so on. And today he's going to talk about world's plants. So Peter, take it away.
Thank you so much for having me. So today I'm going to talk about, I'm wondering whether we could get it darker in here. Is that possible? Great. No, that's great. Okay. Is it too dark when it goes all the way dark? The images are much easier to see. Well, who's afraid of the dark?
No, I think that this is, people will sleep. But if you see a slide, then I'll switch it off.
Okay, fair enough.
Yeah.
So today I'm going to talk about work that is an ongoing collaboration. It's work that I've been doing over the course of the last, I'd say, 7 or 8 years with the Smithsonian and folks at the University of Maryland, David Jacobs, and Neeraj Kumar, who's out there somewhere, I hope. Is an integral part of this, a graduate student of mine who's soon to graduate. And this is work on photographing and identifying the world's plant species. And it's also sort of lessons learned from this process. So those of you who have no interest in plants at all, I'm going to talk about other stuff as well. But I'm going to start with plants. So the 18th century was sort of the age of exploration. And James Cook was one of the great explorers. And he circumnavigated the globe many times. Dying on a beach in Hawaii, killed by the natives. But before he died, he brought back boatloads of specimens, which were studied for their natural science value. And the United States, not wanting to be outdone, also embarked on these great expeditions. And the biggest one was Lieutenant Charles Wilkes, who was given money by Congress to find a hole between the poles, the North and the South Pole. And he never found that hole. But what he did do was to bring back so many specimens from his expedition that they needed a place to house them. And that place became the Smithsonian. So here's an example. Here's a photograph, or many photographs, behind the scenes at the Smithsonian. So if you go to the Smithsonian, the Museum of Natural History, you'll walk around and you'll say, wow, this is an amazingly big museum. But actually, what you see is quite small. Most of it is stored. Most of the stuff that they have is stored in these cabinets, hidden away from everyone else. And a lot of these cabinets, unfortunately, are contaminated. They have mercury contamination because of the preservatives they used over time. The compounds contain that. And so if you want to see one of these species, you're kind of out of luck, right? It's really hard to do unless you're a scientist and you can go there and they'll open up a drawer and they'll show it to you. And so one of the things that we wanted to do as part of this project was to photograph all of their type specimens. Now, the type specimen is a very special thing. The first time a species, let's say it's a plant species, is identified and described, then you can't just say, I found the species and it kind of looked like this and then go away. You have to take a physical specimen. And that physical specimen becomes the type specimen. So it's the reference back that you have to, it's what you have to refer to, essentially. And it has to exist somewhere. And in the case of plants, it exists in a herbaria somewhere in the world. So we wanted to photograph all of the type specimens that the Smithsonian had, and they have the world's largest collection. And so this was an early part of this project, and it was to photograph the type specimens. And so there was a room that was set up in the Smithsonian with sort of a state-of-the-art camera and for this digitization project, and then sort of minute after minute, hour after hour, day after day, week after week, month after month, those type specimens came through. And essentially, well, before I say that, how many different plant species do you think there are on the planet? Vascular plant species. Who has a guess?
100,000.
Excellent guess. About 300,000. So there are many more insects, about 300,000 plant species. And as part of this, we digitized their entire type specimen collection, which is 95,000 types. And this is one of the types over there on the right. Very high resolution. All of these were digitized. They're online now. You can see them if you care about this. So if you're a scientist, you're a botanist, you want to look at this, even if you're not, it's all there. Or you can send away for really high resolution DVDs. It's terrific. And just to give you a size, a sense of this size, it's about a third of the world's plant species, right, that exist tucked away in the Smithsonian as these press types. So, however, while these images are botanically and historically useful, there's just no doubt about that. You just can't do away with them. Or the actual specimens themselves, the images are not very useful for automating the visual idea of species. It just turns out they're not that useful. At least they weren't to us. And species identification itself is a very difficult process. So if you're a scientist and you're looking to discover a new species, you're looking for something that doesn't exist in the herbaria around the world. So, and the way it used to work is that you'd have to get say you're looking for a new species of ginger and you're off in the tropics somewhere, and you think you found it, you'd have to send away to the herbaria around the world and have them send species that you think it might be, and then you'd have to say, no, it's none of these. It's a very exhaustive process. It's very slow. It's very much in the 19th century. And we wanted to make a step toward changing that. Now, if you're not a scientist, if you're an amateur or an ecologist or you just care about plants, identifying species is very difficult to do. It's a very difficult process. And the reason for this is that traditional field guides are impossible to use. I don't know whether you've spent much time with them, but if you take a field guide out into the woods and you stand under a tree and you try to figure out what it is and you have no knowledge, right? So your parents didn't tell you what it was, or you didn't grow up knowing what it was. To find that species in the book is usually a lost cause. If you spend enough time, you can get it, but most of the time you might get the genus, but you don't get the species. So we wanted to change that, and what our goal was to develop mobile technologies for species identification. And so this is something that we talked about, David Jacobs and I talked about 8 or 10 years ago. We had a lot of conversations about this, and I also talked to my son, who has a lot of big ideas, but he was very small at the time. And he's like, Dad, I completely get it. I know this is what it's going to look like. So he went off, and he came back 5 minutes later, and he said, this is it. So he made this drawing. And this is, and it really is exactly what we ended up doing, right? This was it. This was what we did. So he's very proud of that. He never lets me forget this. Dad, are you still working on my idea? He says. So I am, and I'll show you, and I'll get to that. But before, we show what it is that we've been producing, I have to talk a little bit about the technology that's behind it. And so the first thing is that we wanted to photograph living specimens, and the reason we wanted to do that is because type specimens are dead. And they look dead. They look like this. So this is the American beech. It's Fagus granifolia, and this was taken at Rock Creek Park. This was a park near D.C. in 1895. So this is over 100 years old. And there's a lot of beauty to it. I mean, for me, I just love looking at these things. But it does look like a dead plant that's been in a book for 100 years. And we couldn't figure out how to extract information from this to automate species ID. And one of the things that we also know is that people looking at this and then looking at something in the field, it's just hard to make the similarity, right? This thing has been pressed. A lot of them are moldy. Some of them are broken. Leaves are overlapping. It wasn't going to work. If you look at a lot of the images that are in traditional field guides, they're all over the map. Sibley has a nice book. He has a beautiful book on birds. A not quite as beautiful book on trees, where he did a lot of this in watercolor. And it's very hard to tell from the watercolors what it is that you're looking at. And then if you look at books that are based on photographs themselves, here's the American beech again, but it's just the clutter is so great, it's really hard to figure out what it is that you're looking at. So what we decided is that we really needed to begin the process of systematically recording each species visually, in a way that we could capture the living beauty of the species and allow people to look at something that they're holding in their hand and then looking at a mobile device and say, yeah, that's it. And so here's an example. So here's the American beech. Maybe now we could drop the lights for a minute. And so then here are images of the American beech. These are just some of the images that are acquired. And we're working with a group called Finding Species, just a fantastic not-for-profit organization that does nature photography of this type. And so we've been working with them. And so here's just a sampling of the sort of images that we have for American beech, the leaf, the flower, the fruit, the bark, and so on. And then maybe we don't need the lights now. And then, so those are images that were supposedly useful for human identification. But we also need images that would be useful for computer identification. And so it's not enough to have just one or two samples of a leaf. And the reason is that we want this device to be usable by school children and scientists. And you can't tell people how to photograph something without them doing it exactly the opposite way. So we needed many images of leaves, both to experience the variation that people are going to introduce by the way that they hold the camera and the lighting that the leaf is being illuminated by, and also because species exhibit variation themselves, right? So the sassafras can have 1, 2, or 3 lobes, for instance, and so we had to have that variation represented in the data set. And then with that, we wanted to build algorithms that could automate the process of species ID. Now, we knew we would never get it perfect. There's just no way to do it. There's a lot of ambiguity. But what we wanted to do is that when you photograph a leaf, one of the species that it is going to come right to the top, right? Maybe not the top one, maybe not the second, but within the top 2 or 3 or 5 or something like that, as opposed to the top 100 or a top, in the case of the United States, there's 750 tree species. Right? And so if you're just going through a book like this, you're never going to find it. So the first thing is we have to get the leaf off the background. And you'd think, after 30 years of computer vision, that we would have segmentation algorithms that you could just take and use. And it turns out that's not true, at least for our application. Right? There were no segmentation algorithms, and we've tried them all, that would work for our purpose. And so we developed a very simple one, and we also restricted the user to place the leaf on a clipboard with a not uncluttered background. And our algorithm essentially just looks at segmentation, an EM segmentation on the color distribution in the HSV space, and we just use the SV channels. And then we follow it up with a min cut. And so just to look at what that might look like, so here's the first iteration of the EM, the second, and then the third, and by the third it converges. And then we're able to pull the leaf off the background. And the reason we want to pull the leaf off the background, why is there no leaf there? Oh boy. Anyway, there's a leaf there, and it looks like that. Well, wow. Yeah, it looks like that. Okay, so we pull the leaf off the background. And once we pull the leaf off the background, what we want to do is extract information about the shape of the leaf along the contour, which will allow us to tell species apart. And so what we noticed, if you look at enough leaves, and we've looked at a lot of leaves, is that leaves have discriminating information at many scales. So at the finest scale, you have teething along the margin. At larger scales, you'll have lobes, like in the maple leaf. And at very large scales, you can differentiate between simple leaves and compound leaves, which are composed of many leaflets. And so we looked at a measure, an integral measure of curvature over scale. And essentially what it does is it passes a disk along the contour of the leaf, and at each point it computes the fraction of the disk that contains the leaf. And then you subtract a half to get an approximation of curvature. It works very well. Right? It's a wonderful measure because it's an integral measure, and it's much more stable than differential measures of curvature. And I wish that we could say we invented it, but we didn't. Ironically, it comes from the Journal of Molecular Graphics back in 1986, and then it's been reintroduced and formalized nicely by Stefano Suato's group in a 2006 PAMI paper. So how does it work? Well, look at these leaves. So we have 4 different species, A, B, C, and D. And if you look at these, you'll notice that A and B are very similar at a coarse scale, but dissimilar at a fine scale. And likewise for C and D. At a coarse scale, they're very much the same, and at a fine scale, they're different. And then if you look at A and C, they're similar at a fine scale, but different at a coarse scale, and the same for B and D. And so what we use is this representation, which is essentially histograms of curvature over scale. And we look at more than just 2 scales. We look at 20 different scales. It's a very quick measure to compute, and we needed something that's very fast because we want the system to be used by many people all at the same time. So how well does it do? Well, here's how well it does on the trees of the Northeast. So here's a graph which shows that the percentage of time it's correct on the first guess, within the first two guesses, within the first three, and so on. And so it's 70% correct on the first match, and then 96% of the time it's in the top 10. So we'd like it to do better, although you'll see if you use this, it really almost always brings the correct species up onto the top screen. But we noticed in doing this that we sometimes make mistakes that seem avoidable. So, for instance, sometimes we can confuse a leaf which is simple with one which is compound, that's made of leaflets, which is very frustrating because you think that you should be able to get that right. And I'm going to come back to that, right? But this is what's in the system now, and we're working on changing it or making it even better. So now all of this technology is being squeezed onto this mobile app, which is an iPhone app, and also there's going to be an Android app, and we're going to launch it with the Smithsonian on May 2nd of 2011. And so what does it look like? So here it is. It's on my phone. And hopefully I'll give a little demo of this on the iPad later on. So here it is. So this is the app. That's the front screen of the app, and it just kind of scrolls through randomly some of the images that we have in the collection. And then, of course, you can browse all of the images in the collection. So here are the different trees. These are all the trees of the Northeast that are represented on the browse page. And then you can also look at the flowers from the species and the fruit as well. And then you can search. So, for instance, you could search for a magnolia and then choose the saucer magnolia. So here is each species has its own page where we try to represent all the visual characteristics of the species. And so here's the flower of the saucer magnolia. And of course you can zoom in on any of these. They're very high resolution images, and we're just jumping off the app to show you that there's all this detail there. And then other flowers and zoom ins. And here's the fruit. The magnolia has just beautiful fruit. And these gorgeous red seeds, which you can come in on. And petiole, which is the stem. You can even see the hairs on the stem. And then the bark. And we have textual information that describes the species, its habitat, its bloom time, its longevity, and so on. But of course, this is more like a traditional field guide, except the fact that you can reorganize it how you want, and we have images that are very different from what you'd see in a traditional field guide. But the whole point of this is to be able to figure out what you're looking at by just taking a photograph of a leaf. And so New York City I live in New York, and New York City has this program. It's called the Million Trees Program. And what they wanted to do was, in the span of 5 or 10 years, plant 1 million trees to kind of green the urban environment. It's just a really noble goal, and they're actually getting quite close to it. And we lost an elm tree a couple of years ago in front of our house. It's just a beautiful elm, and the crown rotted, and it came down in a storm. And they put another tree in. And it was in the fall, and it still had some leaves that were on it, and they were sort of dried. And I work with trees all the time, and I've cared about this forever, and I couldn't figure out what this tree was. I couldn't figure it out. And I looked in field guides, I couldn't figure out what it was. And this was an early prototype version of the app, and I took a photo of it with the app, and it came back and said it's a sawtooth oak. I said, well, that's clearly wrong, because I know what an oak looks like, and that's not an oak. So that was a little discouraging. But it did it enough that I actually went into a field guide, went into the index, and looked it up, and it is a sawtooth oak. That's what it is. It turns out that oaks don't always exhibit that kind of multi-lobed, pointed shape that you traditionally associate with the black, white, and pin oaks. So it got it right. And so here's how I photographed that leaf again. And so you take a photo. That photo is then uploaded to a server in our lab. And then it's segmented, and then it's recognized. So it basically just ranks in order of what it thinks it is, and the first match is a sawtooth oak. But to confirm that, you can click on the oak and go in, and unfortunately that's a dried leaf, but if the leaf had not been dried, you'd see that there's the leaf there, and a zoom in on that, and then the information about the leaf, and then you can look at the flowers of the oak. And if you're lucky enough to be standing in front of it when it's producing fruit, that's the fruit of the sawtooth oak. And I didn't have that. The tree that they put in is just too young, and it's not producing fruit yet. And then there's the acorn. of the oak, and then here's a blow-up just to show you the detail that we have there. And that's the blow-up on the app. And then finally the bark, and of course the tree I have is too young to really get the sense from the bark. But once you know what species it is, you can just swipe your finger and label it, and then that species is entered into your collection. And then we know where it was taken. So I live in the West Village of Manhattan, and so that's where that photo was taken, on Bank Street, that's my street, and in front of my house. So we record all this information. Now, here's the trick. We're not recording this. We want to record this information so people using the app would, of course, have these collections. But every time they take a photo and they identify a leaf, we get the information back. We store it. And the reason we want to store it is because we want to work with the Smithsonian, trying to map the species across the United States and hopefully beyond if we can get more coverage. Right now we're just in the Northeast. So that we will know where the trees are, we can know what the distributions are, and we can help map the ebb and the flow of the flora over time with the help of hopefully school children or anyone who wants to use this. So New York City, the park systems now wants to use it to do an inventory of the green environment along the streets. But anyway, we'll see. They hope to use it this summer. Of course, all this, for those of you who don't have an iPhone but are still interested in this sort of stuff, you can go to our website, leafsnap.com, and all of the functionality is there, but in a web form, right? And so here's a search on maples, and that's 4 of, say, the 10 or 11 maples that are contained within it. Okay. So this work has been going on for about 8 years now, and it's finally going to be released, and we're really excited about it. But the thing is, I try to ask myself, what did we get out of this? What did we learn from this? And so the first thing that we learned, and it turned out to be really obvious, is that it's really hard to develop technology that can be used by school children, and everyone has a threshold, which is very high for failure. Right, so they just don't like to see things not working. Frustration is, people are easily frustrated, and so we work quite hard to make this as simple as possible. But that observation is kind of trite, and you could have figured that out before you worked on it. But something else came out of this, and it's something that I've been really excited about over the last 3 or 4 years. And that is, in doing this work, I began to look back at some of the ways that species identification had been done historically. Right? Because we're not the first people to think about it. We're just trying to come up with a computational way of doing it. And clearly, people have been doing this process forever. This is taxonomy. And so if you go back and you look at the history, Dioscorides had this wonderful book. It's a pharmacopoeia. It's about the medicinal uses of plants. And it was put together in the 1st century. And it was such a popular book that this book remained in print, if you could call it that, because of course they weren't printing, for hundreds and hundreds of years. And in particular, this is a photograph of one of the pages of that book, from the 5th century, right, done on a lambskin, right? So it hangs around for hundreds of years because it was so useful. And now I also have a friend, Megan Reed at UCLA, who's an Islamic scholar. And when I talked to her about 6 years ago about this project, she said, I really want you to know about this text. It's by Ibn Washia, and it sounds exactly like what you're doing. And he lived in Islamic Spain in the 10th century, and he put together sort of the equivalent of the pharmacopoeia, but for agricultural plants. And so this is a book about the agricultural uses of plants, and it really helped if you look at some of the scholarship around this to advance agricultural production and science. And then finally, here's a book I'm sure you're all familiar with, and this is Linnaeus' Systema Naturae from the 18th century. And if you think about it, what do these books have in common? Well, clearly they all care about living things, but they had something in common which was different to me. And what that was is that each of these works is an effort to teach the reader how to identify an object, in this case a species, by what we call describable visual attributes, things like shape, color, size, and so on. And when we started working on this plant stuff, we took a very straightforward computer vision approach, which is we're going to extract these low-level features, we're going to feed it to a classifier, and then we're going to see how it does. But these people weren't doing it this way. They were doing it a very different way. And so a taxonomy had been built using this notion of a dichotomous key and describable visual attributes. And this method has been around for millennia. So we began looking at this. And so just as an example, here's an image of a leaf. Does anyone know what leaf this is? Maple, good. What type of maple? Close. Sugar. Excellent. Okay, so this is a sugar maple. And if you look at this, if you're standing next to a botanist, actually, if a botanist were holding this in his hand and he was on the phone and you said, describe this to me, they might say it's simple, meaning not compound. It has 5 lobes. It has rounded sinuses. See where it comes in. It's sharp teeth, as wide as it is long, and it's flat. And you could say more about it. But if you said just those 5 or 6 things, and you knew that you were standing under a tree in the Northeast, you'd know you had a sugar maple. Right? So you can get to the species not by extracting curvature along the boundary, but just if you could determine answers to these very simple questions. So, sugar maple. So why is this relevant to computer vision and computational photography? And I think that the reason this is relevant is that it's a different way of thinking about the process of identification. And in the last 3 or 4 years, the use of what we call attributes now is exploding in the field. There's a lot of wonderful work that's happening where people are rethinking the ways in which they do identification. And I'm going to talk to you about some of the work that we've done here, and hopefully you'll see some relevance to comp photo. So why attributes? You know, what's an advantage here? So attributes are composable. And because they're composable, you can describe object categories like white man. You can describe objects like white man with blue eyes, brown hair, and scar on cheek. And you can also describe instances of objects. So you're reusing these. You can say white man, blue eyes, brown hair, scar on cheek, looking left, lit from the right. They're generalizable. So you can describe things you've never seen before, like women with mustache, or not seen too often, like smiling Asian toddler with glasses. And they're efficient, right? So in theory, K attributes can describe 2 to the K categories. In practice, it's a lot less than that. But in theory, it's 2 to the K. So they're very powerful because of their efficiency. And here's the claim. Within certain domains, it's possible. And so I'm going to show you one of these domains, and that's the domain of faces. And this is work that we've been doing for 3 or 4 years now. And so let's look at that. And so the idea is we wanted to see if we could build classifiers that could look at images with faces and very quickly determine what attributes the face possessed. And we realized that we didn't want to just do it for gender, and we just didn't want to do it for smiling. We wanted to do it for a whole vocabulary of things. And if we're going to do it for a whole vocabulary of things, it had to be automated. You can't possibly sit there and make these things by hand. You have to set up a pipeline, and the pipeline has to be automated. So much so that the only thing you need to do is to come up with the vocabulary. And then if you do, then you can train your classifiers by getting data by downloading images from the internet, taking those images, spitting them out to Mechanical Turk, which is this wonderful service for getting users in the loop, asking them to mark images if they exhibit the attribute or don't exhibit the attribute. And then once you have your data, you can train classifiers. And we set up this pipeline to do it. Originally, we did this using graduate students at Columbia before the SIGGRAPH deadline. And we got in so much trouble for that. And because we promised, we said, if you can do this, whoever gets the most, we're going to buy them an iPod Touch. That's what, and then the next person is going to get, what was it, Niraj? An iPod Nano. So we started this, and it was incredible. We just got all these labels start coming back, were coming back to us. But professors were starting to get annoyed. But what we did find is that a lot of the work that was being done was being farmed out to relatives in China and India. So it became, it's this sort of an early version of this Mechanical Turk. And so we later switched over and started using Mechanical Turk, and we were able to label a lot of images. But before you can label images, you need them, right? So what we did is we set up a crawler. Niraj did this. We started collecting image after image after image, and so much so that we had over 3 million images, and we just had to stop. So you download images, you use face detectors, you find the faces within the images. And this data set sort of dwarfs sort of previous face data sets that are out there in the face recognition and verification community. And I've been, I was responsible for Yale A and Yale B. So, and then the ferret and the CMU Pi and FRGC are just little things compared to the size of this data set. Every pixel here is 10 faces. There are 10 faces there. So, and the second thing I wanted to say is that all of the faces in these images are faces in the wild, a term that comes from Eric Leonard Miller, who started the labeled faces in the wild data set, a wonderful data set, and sort of inspired by that. And they're not unlike our databases at Yale or Ferret or CMU or any of these other databases, which were essentially gathered in the lab. These are images that are just out there on the internet. People aren't behaving. They're lit in any way. They're expressions or whatever. They're looking in any direction. They're just faces in the wild. It's the sort of stuff that you would find on a cell phone camera. And then we used mTurk to label, to get more than 1 million labels. Was it 1 million? It was about 1 million. About 2 million? Okay, about 2 million labels. And it cost us to get 2 million labels about $10,000.
Less than that.
Less than 10, like $5,000 to $10,000. This doesn't cost very much. It turns out there are a lot of people that actually enjoy doing these jobs, right? It's not. No, it's just. It's like doing a jigsaw puzzle or like a word search. They get to look at faces and then just say, is this a man or a woman, or is she happy or sad? And they'll do it. But to build a classifier, you need to get features out, and we wanted again to do this in an automated way. And we knew that the features which were going to be useful for hair color were probably not the same features which are useful for telling whether someone's smiling or what their gender was, and so on. So what we wanted to do is to set up a palette of possible feature types and regions on the face that the classifier could select from automatically. So different regions on the face, different types of image information. And then we would build a classifier that essentially selected what was most useful in a greedy, iterative way. So let's say in the case of gender, you had training images, the positive examples say females, and negative examples say males. You extract low-level features, and then in a greedy way, you extract the features which give you the best classifier for this task. And once you have this classifier, once you train it and you have this classifier, then you could submit to the classifier images of faces, and it can tell you, say, what the gender is, right? And not just tell you that, it's almost like a regressor. It can give you a measurement, which is the degree to which it thinks the face exhibits that attribute. So this is not, maybe this isn't appropriate. What's his name? My daughter loved him. Orlando Bloom. He kept coming up as a woman. But because he has long hair and there's a whole thing going on there. But anyway, so attribute class. So we did this for a lot of for attributes. So here are the attribute classifiers and their accuracy. So you can just see it's just on and on. I think we have 70 plus, Niraj? 70 plus. So this is just one page of them, and here's another. And so once we can do this, we thought, why can't we just use this to do image verification? So instead of looking at low-level features and comparing the low-level features, we could compare the attributes, these sort of intermediate-level features. Clearly if someone's flagged as a man and someone's flagged as a woman, they're clearly not the same person. And so a big problem in face recognition and verification is to be able to tell whether two images contain an image of the same person or different people. That's the face verification task, and we applied the attributes to this problem. And in this case, it's an easy one. They're obviously different, and the classifier would get that right. And here are results on Eric Leonard Miller's labeled faces in the wild, so we're the top result there. So interestingly, using these features, which are these attributes, which are essentially taken from the taxonomy literature, we could produce state-of-the-art results on a very, very challenging data set. Now, you can do an awful lot more with them, though. And so one of the things that we were looking at, actually before we did the verification work, was to see whether or not we could use the attributes to do searches in image databases. So this is a search from Google about 3 years ago if you typed in smiling Asian men with glasses, basically random stuff would come up, including an image of cucumbers. And it's just there's nothing there. I mean, there is an Asian man there, but he's not wearing glasses. And if you search the data set that we had put together, this is what would come up, right? So you could find the smiling Asian men with glasses, but you could also search for, say, young blonde women, or you could search for older men with mustaches. Sorry about the losing contrast there. And then here's a search for dark-haired people with sunglasses. And there's a little fallout. And then here's a search for attractive, influential vision researchers. So, I mean, it works. Essentially, it works. And then an application that we wanted to do. I have my wife's father. Is just a wonderful man, but he at Thanksgiving, what he does is he wants to take a family shot, and of course that's a great idea because you're happy about it after it happens. But there's about a 20-minute period where it's actually very painful, because my kids will never behave. Right? They close their eyes, they frown. It's just a mess. And there's some nice work on doing that's been going on in the photo literature and doing this sort of stuff so that it's photorealistic. And we also had a paper at SIGGRAPH in 2008 which allowed you to replace faces so that you could basically automatically take the face from one person and insert it into the face of another person and have it look actually photorealistic. However, what we thought is that you could also use this attribute stuff so that you could have the camera make a composite photo that had all the qualities that you wanted. So in my case, or my father-in-law's case, it would be that everyone was smiling, and that's very important to him, and that their eyes were open. And so I had my daughter sit down on a sofa with two of her friends, and we took a bunch of photos. And so here's what they look like. And so I selected some of the worst ones. And if you step through them, you'll see that her friend Claire's eyes are closed, and then George is frowning, and then my daughter, she closed her eyes. She wouldn't close her eyes, so I said, just close your eyes for the shot. And then finally, George's eyes are closed in the shot here. And so what we thought is that if you have this image stack like this, and you can replace faces in a way that's photorealistic, and you can tell what someone's expression is and whether their eyes are open, then it's very easy to make a composite. So here's a composite photo, right, where it satisfies eyes open and everyone's smiling. So that's using attributes for a different purpose. Now, in addition to this, there's a lot of wonderful work that's going on in the vision community now, looking at the sister of attributes, which are parts. So trying to find object parts. Tenra Malik's group is doing a lot of nice work in poselets for this, and I've always looked at faces. So Neeraj and I have been looking at ways that we can isolate parts of faces, right, using a lot of the same ideas. And we wanted to be able to do this for faces which are in the wild, people that are looking not at the camera, where there's partial occlusion, where there's crazy expression, where the images have low quality. And Neeraj is going to have a poster that he's going to show on that on Saturday, I guess. And this will be a paper at CVPR. And these are just some examples of the algorithm working. It's fully automatic. So it can find the mouth and the nose and the eyes and the eyebrows, in spite of occlusion and very low quality images. And then finally, before I give a quick demo, this is very nascent work. We've just begun it. We want to use the same sort of ideas to look at automatically labeling images of architecture. So I'm very interested in this. Obviously there's this wonderful data, and that's Google has Street View, and so you have images of all the buildings. And I'm very fascinated with the architecture in Manhattan. And Luc Vincent, who's one of the sort of runs that program, gave me the nod that it's okay to do this. I don't know whether he's going to change his mind. But what we did do is we went in and we extracted images from Google Street View. So that's just a raw image, and that's an image that's been rectified. And then we've taken all of the images in the West Village, which is essentially 1,000 buildings. So within this region, we have all the buildings. And all of those buildings we've extracted And not only have we extracted them, we've had people manually label them. So we have labels for everything. We know the windows, the facade, the cornice, the sky, the trees, the stairs, the doors, and so on. And what we've begun to do is to use this information to build detectors and classifiers and automatic labeling systems that can find buildings and pull out the parts automatically. So here are very preliminary results on this. And we're really excited about this because I really think that in not too long from now, we're going to be able to unleash this across all of the images of Manhattan and be able to find these buildings, find their parts, and then find matches within this data to other buildings which we have architectural and historical information from that we can share and begin to give descriptions about what it is that someone might be looking at. And we'd love to put this on a camera, on an iPhone, so you could just walk around, take a photo, and it'd say 1856 Italianate townhouse, with architectural references and so on. We're not there yet, but I think we have the building blocks in place, and we're really excited about it. So now, Srinivas, if I could, I wanted to go and very quickly show the iPad version, which is not actually ready, so it probably won't work, of the app. And the reason I wanted to show this is because we can get, let me turn this off. Blank screen. Is we can get, it just has so much more screen real estate, and also has 1080p out, so I can show it. It's there. Oh, yes. So, Srinivas, can we kill the lights entirely? So I'm just going to stand here. So here's a very not ready version of the app. And this is on the iPad. And it's pushing out. So this is just the front screen, and then there's all information about it. So what I'll just show you is images. So if I go here, let's go to that bur oak. That's one of my favorite. So here's the bur oak, right? So you can select that, and you can really see it in all its glory. And then you can look at, say, its leaf. I mean, its flower. Really, you get a sense of what's there. And then flowers, and then you have the fruit. All right. So there you go. And of course, this is about where you can get information about the species, right? But this is about recognition. I don't know whether I have an internet connection. So if this doesn't work, I'd like to conclude the talk immediately. But we'll see. So I don't have a leaf with me because there are no leaves on the trees, but I just took some from my collection. Oh, darn. Let's see. Let's see. Okay. There. Please have an eye. So it's uploading the image to the server oh, there it is. Good. Okay, so that's the London plane tree. So you could, so that went to Columbia in my lab, and it's recognized it. And then you can go in, right, and you can see all the information. It's just a beautiful plant. There's the fruit. And a lot of you probably know this as a sycamore, right? And so here's a bark that you might recognize. But anyway, so that's the app working, and of course you can do it for. Here's another one. I hope this works. Oh, sorry.
How many iterations of the interface did you go through to get to this? This is very elegant.
About 5,000. It really is very sad. I would never do this again. It's been fun, actually. We've been working with some amazing students, and a lot of students are doing this as a master's project. Here's one of my least favorite trees. This is Atlantis, the tree of heaven, which is an invasive species. There it is. And you probably recognize it. It has a very pungent, to say it politely, flower that blooms in July. Anyway, so this is the app. The iPhone version will be out on May 2nd, and the iPad version hopefully in the summer, and it's all free. And so any of you who have interest in plants, and even if you don't, just download it and say nice things about it. Thanks so much.
Did you have to do anything with color for compensating on the devices?
So that's a really good question. We didn't. I mean, the color on the devices are just beautiful, and I think they spent a lot of time trying to get that right. Apple, I guess. The color on this projector is slightly problematic, but on the device itself, it actually looks a lot better. Yeah. Yeah.
You're not using any color feature or internal structure feature of the leaves.
So yeah, right now we're using just the external shape. Although we have work on looking at the venation, right, the kind of the vascular structure within the leaf, which is not ready for prime time. But it's a harder thing to extract with a cell phone image taken by a child, and easier to extract in the field. And in particular, a lot of tropical plants are discriminated based on venation patterns. So we've looked at doing this for an outpost that the Smithsonian has on Barrow, Colorado Island, which is an island in the Panama Canal, where they're doing a very extensive study of tropical species there. But on the app, no.
How do you take care of the...
So on the results you were showing of the searching of like Asian man with glasses and things, were those labels from the mechanical Turk labeled set, or were those labels labeled automatically?
Oh, those are automatic. Yeah.
Okay.
Yeah. Those are it's fully automatic. Yeah. Yeah. Oh, the flower is so great, and there's been actually Andrew Zitterman has done some work on recognition from flowers. You know, I have a lot to say about that. I'm not sure people would be interested, but the problem with doing identification based on flowers is that plants rarely flower, right? And so if you're standing in front of a tree or a woody plant, and you want to know what it is, there's only a really narrow window that it's going to be flowering, and some don't even flower every year, right? Some flower every 20 years. So yes, and then the second thing is that flowers, the color of the flower is very discriminative, and flowers have a color signature which we're essentially blind to. A lot. So, for instance, there are flowers that all look white to us. They're metamers, color metamers, but to bees and birds, they're actually very different colors, right? But you know, you don't have these aren't fancy multispectral devices, so we really couldn't use that information. Yeah.
You had a question queued up before you had the anecdote about shooting outside in the West Village, but do you use any other metadata like the location of where the photo is being taken to say this tree is unlikely to be in this state?
Oh, absolutely. And so not now on the app. I mean, the recognition I think is good enough for the Northeast that we don't really need to winnow in that way. But we're going to go across the country, and certainly it's true that if you're standing in Florida, you're not going to see a sugar maple. And so we know what states they're in, and we just haven't put it in because we don't need to. It's really simple to do it, and that's exactly what one should do. Yeah.
What's the classifier?
Pardon?
What's the classifier that you use to read out what's...
For the leaves or for the faces?
For the leaves.
For the leaves, we're using a nearest neighbor on a chi-square distance. Do we change?
I think histogram intersection.
Okay, now it's histogram intersection. Yeah. For faces, it's a support vector machine with an RBF kernel.
How big is your database that you're using to do the match?
For leaves? Well, we have about 360 species, 200 of which are trees. And we have, each tree might have somewhere between 4 to 20 field images that were taken of leaves from it.
But I mean, in terms of like memory storage, like how much data is this thing going through when you do the real-time match?
Oh, it's not very big at all, right?
Yeah,
You know,
I mean,
could you store it on your phone? That's what.
Oh yeah. So we could do the recognition on the phone, and in fact, we've done. It might be a little bit slower, but the reason we don't want to do that is because if we did it on the phone, we won't get the data, and we want the data. So we want to force people to talk to our server. Yeah.
Is there a work plan for extending this to mushrooms?
Yeah. I can't believe you asked that, because David Jacobs, who's is the colleague working on this, the collaborator, said, why don't we do this for mushrooms? That was his first idea. And my reaction was, we will be arrested, right? I mean, for all the obvious reasons, right? Including, like, someone, we say, oh, it's a, a harmless whatever, and then... So I think it would be really cool, but I think that we would have to have a waiver, like this long. But I would love that, Any other questions? Well, thank you so much.